According to the USDA's National Agricultural Statistics Survey, we raise 92 million cattle in the U.S., providing about 208,000 pounds of milk and about 24 billion pounds of beef per year. Some of the most common illnesses that affect dairy cattle and calves are mastitis or inflammation of the udder, metritis, inflammation of the uterus, and pneumonia. There are specific times during their life when they are most at risk of contracting bacterial infections: at weaning, shipping, and when commingled with animals from other herds. For example, bovine respiratory disease complex is a major threat to their health and welfare, and a variety of viruses and bacterial pathogens contribute to disease. Antibiotics are often prescribed by veterinarians to treat, control, or prevent these diseases, although some antibiotics are also sold over the counter. Some antibiotics are also added to feed or used routinely to promote weight gain.
The first critical control point is the use of antibiotics in livestock. And the question is, do antibiotics select for antibiotic-resistant bacteria in the manure? And we're looking at both beef and dairy cattle and the common antibiotics used. We're looking to see if the antibiotics used actually select for resistant bacteria, and we'll be looking at the DNA and a new method called metagenomics, which is going to allow us to profile all the different kinds of antibiotic resistance genes present in the manure.
Among commonly used antibiotics in the dairy and beef industries, macrolides, one class of antibiotics, are categorized by the World Health Organization as critically important in human medicine, while cephalosporins, sulfonamides, and tetracyclines, three other classes, are highly important. In this project, representatives from these antibiotics classes were dosed to dairy and beef cattle in accordance with common practice and compared to non-dosed controls. Manure and urine samples were then collected regularly. The manure samples were then analyzed for antibiotics, antibiotic breakdown products, antibiotic-resistant bacteria, and antibiotic resistance genes. This study will provide guidance on manure management strategies for minimizing the spread of antibiotic resistance and whether or not antibiotic use has a measurable effect on persistence of antibiotic resistance genes in the manure.